For Gabriel, time is running out. His muscles are getting weaker and weaker. His legs have already given out. At some point, his heart and respiratory muscles will follow suit. But suddenly there's hope for Gabriel. Geneticists have developed a new technique called CRISPR. It's like a pair of scissors that can edit genetic material with great precision. It holds out the prospect of completely new therapies, not just for rare genetic diseases, but also for cancer, HIV infections, and many other disorders. The elevator in the living room makes daily life easier for Gabriel. But he knows that his life is dependent on medical progress.
CRISPR is going to change and probably even revolutionize the way how we are going to practice medicine one day.
And not only medicine. The CRISPR technique can also give plants new characteristics, resistance to pests or drought. Even some organic farmers see opportunities in genome editing and say they should be exploited. Critics, though, are warning about a new era of genetic engineering because gene editing makes possible previously unthinkable interventions into the genome.
It's certainly going to change our society. And that's why not just scientists, but society itself must address the whole issue of genome editing.
How far should medicine be allowed to go? And agriculture? What are the opportunities and risks behind CRISPR? Gabriel lives in London. He's mad about horses. He spends every free minute at the riding stables.
When I ride, because obviously I can't run around, it gives me my legs back. So when the horse is running, I'm running as well, and it gives me that peace of mind that, you know, that it makes me normal for once.
Gabriel knows what it's like to be normal. As a young boy, he could walk just like his siblings. But at some point it became clear that something wasn't right. He was 4 when his parents got the diagnosis, Duchenne muscular dystrophy. Roughly 1 in 5,000 boys is born with the disease.
Well, at the time we were given the diagnosis, told very pragmatically your son would lose the ability to walk at around 11, and then die in his early 20s, and there was nothing that could be done. And we left the office with that knowledge.
The prognosis proved correct. Despite all the efforts he made, Gabriel gradually lost the ability to walk. At 13, he got his first wheelchair.
It's ridiculous. Why should someone like me have to struggle unnecessarily when I'm practically right at the beginning of my life? So, no, sometimes I see it as like a 90-year-old man in a 15-year-old's body.
Gabriel takes a lot of medicines every day, but they don't hold out a cure. They can just delay the progress of the disease. It's about gaining time.
Every week that passes, every day that passes, I see the difficulty of lifting a glass of juice to drink, brushing his teeth. You know, the difference of the child I'm caring for now and the child I could be caring for in 6 months' time is drastic. And that is why that pressing need to do something sooner rather than later is overriding.
Gabriel's parents have read everything there is to know about Duchenne. It's not that much, because the pharmaceutical industry is not particularly interested in such rare diseases. The Rosenfelds took action themselves. They set up a foundation, invested their own money, collected donations, and established contact with scientists around the globe, including Ronald Cohn, pediatrician-in-chief at the renowned Sick Kids Hospital in Toronto. Cohn is one of the world's few experts in rare muscle diseases. His research involves the development of new therapies. And for the first time in the history of Duchenne muscular dystrophy, there are prospects of a cure. Ronnie, as everyone calls him, is both a scientist and a pediatrician. Nowadays, his research work doesn't leave him too much time for patients, but he still looks after serious cases himself.
Your blood pressure is just fine. Did it hurt?
That's the best one.
Ronnie knows how to allay children's fears. For over 10 years, he's also been following Gabriel's development. Over time, the contact has grown closer. The Rosenfelds and Ronald Cohn have become friends. In the meantime, Cohn supports the Rosenfelds Foundation as a scientific consultant.
Hello?
There we go.
Can you see us?
Can you see me?
Yeah. Yep.
This time, it's not about medical matters. Gabriel wants to talk about his forthcoming holiday.
Gabriel, tell me, what are you going to do at camp?
Probably, swimming, speedboat.
Speedboat, all right.
He's already excited.
Ronnie is more to Gabriel than just a friend. He's the doctor who could save his life.
All right, lots of love, eh?
Bye.
Ronald Cohn is originally from Germany. He's been working in the US and Canada for over 20 years. For a long time, the investigation of muscle disease produced no breakthroughs. Then a scientific discovery made Cohn sit up and take notice: CRISPR gene editing. It offered the prospect of tackling the muscle weakness directly in the patient's cells. A therapeutic concept for many other diseases as well, so far considered incurable.
We are for the first time able to develop a concept of fixing genetic mutations, genetic abnormalities. It's possible, we can think about it, and that's why I think it's one of the most important discoveries of the last few years.
This woman helped spark the revolution in the genetic laboratory, Emmanuelle Charpentier. Her discovery of the CRISPR gene editing process in 2012 has earned the French scientist countless awards. She now heads the Max Planck Institute for Infection Biology in Berlin. Here, Emmanuelle Charpentier continues to work with the bacteria to which she owes her scientific career. In bacteria which cause scarlet fever, she discovered a mechanism with which the bacteria protect themselves against viruses. A molecule that functions like scissors and can cut genetic material precisely, CRISPR. Charpentier had the idea of using this molecule to create a universally usable tool.
What is the most fascinating is to have had the privilege to identify a mechanism that is working in a so neat fashion in nature, and that was obvious to be able to harness as a powerful technology. And it shows really the power of evolution in life.
Our genome is rather like the book of life, an incredibly long text with billions of letters, but sometimes you get typos, mutations, and they can lead to diseases. This is where CRISPR comes in. The molecule works like a pair of scissors with an address label. It looks for a specific text, docks on, and cuts out the typo, or alters the text. Or inserts something else. More easily and precisely than ever before.
It's an easy programmable system. It's more efficient. and it's also more versatile. So one can perform different types of changes on the genomes. So for the biologists, this is really a very powerful tool. It's like a Swiss Army knife.
Geneticists around the world are exploring the possibilities of the new tool. CRISPR works with plants, animals, and humans, and so could fundamentally change agriculture as well as medicine. For Gabriel, and for many other children with Duchenne muscular dystrophy, CRISPR could be the salvation, because the cause of their illness lies in the genes. Gabriel's parents do everything they can to make his life easier. They're hopeful and at the same time anxious about the potential of the new gene editing technique. For the first time since the diagnosis, there is something approaching hope, a vague prospect of a cure.
Science and technology has moved at a pace that is unprecedented, and things that used to take 2 years can now be done in 6 months. So the progress has been incredible. I'm slowly getting to a very uneasy point where actually this could impact my own child.
For Ronald Cohn, too, a treatment for Gabriel is moving within reach. In Toronto, he's conducting research into Gabriel's muscle cells. The muscle weakness is caused by a gene which contains the blueprint for dystrophin, a protein that normally stabilizes the muscles. The problem is that the dystrophin gene is the longest in the human body. It's made up of 2.5 million individual letters. 2.5 million letters have lots of room for typos. Gabriel's mutation is relatively simple. A certain DNA section occurs doubled. Cohn has programmed the gene scissors to cut out this precise section.
We put the CRISPR-Cas9 into the cell culture, waited about 12 days, I think, and there it was. And that was one of these eureka moments.
An initial success, but a cell culture is a very different thing from a human being. How would Gabriel's already damaged muscles react to the treatment? Even the nature of the therapy is still completely undefined. The difficulty is getting the CRISPR genetic scissors to the right spot. Would an injection into the bloodstream be best, or directly into the muscles? And above all, what side effects might Gabriel face? All this is totally unknown.
If I have a boy who already sits in a wheelchair, and I know he sits in a wheelchair because he already has a lot of muscle tissue replaced by scar and fat tissue, is there still enough muscle left that any kind of therapy that could target the muscle would be successful enough to at least keep the boy at the stage where he is and not have him progress any further? And I'll be honest, the answer to this question will only come from clinical trials.
Jesse Gelsinger was a boy too when the latest findings of the geneticists gave him reason for hope 20 years ago. The fate of the 18-year-old American shows that we must be wary of expecting too much. In 1999, Jesse was one of the first patients to be treated with a pioneering gene therapy. He was given an infusion of genetically altered virus particles in a bid to cure his metabolic disease. But his immune system collapsed. 4 days later, Jesse Gelsinger died, a supposedly unforeseeable accident. It only emerged later that the same complications had occurred in animal experiments, but no one had drawn the necessary conclusions in time. Jesse's death put a stop to the development of gene therapy for many years. Ronald Cohn knows exactly what happened back then, and he's determined not to repeat the mistake.
If something even close to what happened 20 years ago to gene therapy is going to happen to CRISPR, it will again paralyze the field for years, and nobody wants to go down that path.
When is the right moment to risk a therapy? Not before all risks have been investigated? But won't that be too late for Gabriel? For Gabriel's doctors, it's safety first. But his condition deteriorates with every day that passes, and lost muscle tissue can't be replaced.
So these are very crazy scenarios to be in. To do nothing isn't really an option, and to do something comes with a whole load of excitement and complete fear.
Gabriel's life is not immediately threatened. He has a lot of things he enjoys. Would he go along with a potentially risky gene therapy experiment?
I would, because I'm desperate to be normal like my friends, to be able to just walk out the house as opposed to, you know. I like to go for a run with my friends. I like to play football properly with my friends, not in my wheelchair, because I don't like being the goalkeeper or the referee. Obviously, my friends are trying to be nice, but that's not how I see it.
But the debate about the opportunities and the risks of CRISPR in medical research is broader than whether individual patients like Gabriel could be helped. While we are filming in London, we hear about completely different experiments involving genome editing. In the US, scientists have changed the genetic material of human embryos. And that's a totally different dimension. Because when researchers manipulate these early stages of human life, the changes will be passed on to all subsequent descendants. Mankind is altering its own germline, the bond between the generations. The researchers in the US were trying to find a cure for a hereditary heart disease. These experiments took place only in the cell culture, but they split the scientific community apart. Emmanuelle Charpentier knows the power of the CRISPR mechanism she helped to discover, and she rejects any intervention in the human germline.
It's an ethical issue. This is to protect issues of human integrity, to not cross certain barriers. And also with the idea that the CRISPR-Cas9 technology is really here for therapeutic purposes and for research and development purposes, and to treat patients, not to treat germlines.
In Germany, such research on embryos is prohibited by particularly stringent laws. But in the US, in China, and in other countries, scientists are continuing with these experiments. It seems it's no longer a question of whether or not a CRISPR baby will be born, but when and where it will be born. In the Berlin Museum of Medical History at the Charité Hospital, we meet Peter Dabrock. He's a theologian and head of the German Ethics Council. CRISPR has made so much achievable that in the past was very difficult or simply impossible. So the ethical questions raised are all the more urgent. What should mankind be allowed to do and what not?
Es ist ein Menschheitsthema.
It's an issue for the whole of mankind, so it can't just be addressed by scientists alone. It cannot and must not be the case that scientists alone establish realities. We need instead a broad, global discussion.
CRISPR is capable not just of treating disease. With it, genetic research could focus on blue eyes, blonde hair, everything that lies in our genes. Since the advent of genetic engineering, there's been talk of designer babies. That could now become a reality. Who wouldn't want healthier, more intelligent offspring?
Do I think that parents would want this? Do I think that human beings would want it? Absolutely. Which is why you need to regulate this somehow. You need to regulate the technology in a way that you continue to enable innovation and progress, but at the same time limit people from doing things we don't want them to do.
But how can you limit people in widely differing countries and cultures from tampering with the human genome, from intervening in human evolution? Thanks to CRISPR, these questions will be with us for a long time to come.
I do believe that our children's world will be a CRISPR world, and that genome editing will shape all life science, medical, agricultural, and technical developments to the extent that the technical world we know will be turned upside down.
While the medical sphere wrestles with the limits of the possible, in agriculture, the gene editing technique has already begun to change the world, especially in the United States. Johnston, Iowa, is the headquarters of DuPont Pioneer, the biggest agrarian multinational. We are among the first television crews to be allowed to film here. The research focus here is above all on the corn genome, also called maize. Every corn plant in the test greenhouses carries a barcode and is fed completely automatically. The big question here is which genes are responsible for which characteristics. As soon as the company knows that, it will be able to produce targeted changes in the genome of the plants to improve their resistance to pests, for example, or to raise yields. Since the CRISPR gene editing technique was discovered in 2012, plant breeding at Pioneer has also become faster and less expensive. The company sees huge business opportunities.
For me, this is the realization of 30-plus years of hope and dreams for how we could improve the food supply. So we already are building a pipeline of product opportunities that will improve, as I said, sustainability, water use, disease resistance, and as I said, we're also working in a number of crops. So I think the next decade will be a very exciting time.
Pioneer is not alone. Many other companies and universities are already using CRISPR to produce plants with new characteristics. Laboratories have already created mushrooms that last longer. Soybeans with a healthier fatty acid composition. Wheat that's resistant to certain pathogens. And potatoes that produce less carcinogenic acrylamide when fried. Pioneer also granted us access to a trial field where the first CRISPR plants are growing, a release experiment that at the moment would be unthinkable in Germany. The plants are waxy corn, which has a special starch composition. Nothing revolutionary in itself because it already existed before CRISPR. But waxy corn will be the first CRISPR product worldwide to come onto the market.
You'd see it a little bit in food, you'd see it in industrial uses. In fact, if you have paper that's a shiny surface paper, it's probably using a specialty starch from our waxy corn. The yield, the performance of this product will be superior to our current versions of the waxy product, and it's because we can just go in and make the targeted changes directly in our best varieties. We can actually deliver really, really good performing varieties to our customers.
The research director is enthusiastic, but the CRISPR corn is also raising old misgivings. Once again, it's the major companies that are driving genetic engineering forward. When the first genetically engineered plants were developed in the 1990s, companies like Pioneer and Monsanto promised they would produce plants that would be good for the environment and healthier for consumers. So far, these plants have failed to appear. Instead, genetic engineering has helped to accelerate the industrialization of agriculture. Monocultures and the use of pesticides are on the increase. Around 80% of all genetically modified plants today are resistant to a herbicide. The seed companies sell sprays against weeds along with the seeds, a lucrative business. And that's what's sparking protest. Monsanto and genetic engineering have become bywords for all evils. Campaigns have led to public opinion largely rejecting what's known as green genetic engineering. The demonstrators are protesting above all against the power of the major companies in the US as well as in Europe.
That's a huge problem, especially in so-called third world countries. I know of countries where, for example, farmers no longer have the choice of not buying Monsanto cotton or Monsanto seeds. From that moment on, because seed, after all, is the starting point of production, they're dependent on a few globally operating companies. As farmers, we can't be neutral to that, and as consumers, we shouldn't be neutral to that either.
So far, genetic engineering has principally been employed where it pays, for soybeans and corn, rapeseed, and cotton, the so-called cash crops that earn companies billions. Are the new CRISPR plants also mainly strengthening the power of the multinationals? The distrust they're facing is certainly great. Prince Felix zu Löwenstein is the chairman of Germany's Association of Organic Food Producers. He represents a totally different approach to agriculture, which rejects any targeted intervention in the genetic material of plants.
I believe that the tunnel vision concentration on the genome is wrong. We must look at the agro-ecosystem as a whole, instead of trying to repair this system so that we can keep it going with all other negative effects, for example on biodiversity. We would do better to change the system itself, by looking at mixed crops, for example, and the development of soil fertility. That would take us very much nearer to solving the problems that we're now trying to solve in the genome.
But are, in fact, a more sustainable agriculture and CRISPR gene editing really mutually exclusive? Is CRISPR simply a new instrument in the hands of the multinationals? At the University of California in Davis, we meet an unusual couple. Pamela Ronald is a plant geneticist. Her husband, Raul Adamczak, is an organic farmer. UC Davis is one of the world's leading agricultural universities. Organic farming and genetic engineering, conflicting fields, you might think, but they needn't be if they pursue the same aims.
We both have a vision, a very similar vision for sustainable agriculture and how the healthiest, most productive food can be grown in the most environmentally friendly way. So, yeah, there's a definite overlap there.
On the university farm, Raul cultivates beans and other vegetables without using chemical fertilizers or pesticides. But some problems are persistent. Again and again, pests destroy his crops. Raul is one of the few organic farmers prepared to talk in public about how new plants from the gene laboratory could help.
Agriculture needs to use most advanced technologies along with the best ecological farming practices. If we can use both our plant breeding brains and our farming practice brains to put together a more diverse and more productive agriculture, I think that bodes well for the future.
Pamela Ronald is a geneticist and has specialized in rice, one of the most important food crops in the world. The multinationals are not very interested in genetically modified rice. They can't make much money from it. because it's mainly grown and eaten in poor regions of the world.
What I became drawn to working on agriculture is to help contribute to an ecologically based system of farming and to help nourish people that don't have much to eat. So there are many people that lack food security. So those were my major reasons.
She's had great scientific success using previous genetic engineering methods, but the work on a new plant variety was immensely expensive and took decades. So recently, Pamela Ronald and her colleagues have also been concentrating on CRISPR genome editing. The researchers hope it will help them reach their goal more quickly and will make varieties possible that were previously unimaginable. They aim to develop varieties that certain pests can't damage. That would protect crops and save on pesticides. In addition, they want to modify rice plants to make them better able to cope with climate change.
A dream would be that we could use this approach to develop drought-tolerant rice that could also be applicable to other crops. That's a big challenge in plant biology right now, especially in the face of the changing climate, is to develop crops that are more resilient to climate change.
Her husband is also looking to the CRISPR technique for help. The many discussions with his wife have rendered the organic farmer open to something new. A root pest is eating his vegetables, especially the squash.
I mean, there's just nothing there.
Yeah.
And that should be a well-rooted plant.
But then look, they want you to pick up the...
If the squash grow at all, they're very small. But there are some plants that have resisted the pest attacks well.
So if we could find a crop, identify the resistance in another crop, then you could put the gene into this crop. That might work.
Or, you know, you saw how the plant didn't produce a lot of roots. Well, maybe you could find some genes that would double or triple or quadruple the amount of roots produced, and the plant could just outgrow the symphyllins.
The two of them want to launch a research project on this pest because organic agriculture has no means of combating it. The toxic substances used in conventional agriculture are taboo for Raoul. He sees a plant from the CRISPR laboratory as the best solution.
I would plant it, but the big caveat is, would the USDA National Organic Program allow me to plant it? So if they can accept CRISPR-mediated plants, then I would plant it, yeah. It's a no-brainer.
What's so obvious to Raoul is highly incendiary. Opposition to genetic engineering is a central pillar of organic farming. We asked Prince Felix zu Löwenstein whether the new CRISPR technique could change that.
These new genetic techniques we're talking about share at least one risk with the old genetic techniques. They involve replicable living organisms, which we are releasing into nature from where we can never extract them again.
True. But hasn't traditional plant breeding been doing the same thing for thousands of years? It's not only the CRISPR corn growing in the trial field in Iowa that's radically different from the original natural corn plant. Most corn varieties were developed without genetic engineering. Through repeated crossbreeding, they're now far removed from the original wild plant, which is also here. Teosinte is the name of the wild, distant ancestor of corn. The kernels are tiny. It's still found in Mexico and neighboring countries. Through crossing and selection, corn has radically changed in the course of thousands of years, and the genome of the plant has been modified completely without genetic engineering. So is genetic engineering nothing other than a form of plant breeding? Well, not quite. because at least with genetic engineering to date, man has gone one step further. To genetically modify corn or other plants, researchers generally use bacteria which contain the desired gene. The foreign gene is implanted into the genome of the corn. A plant modified like this is called a transgene because it contains DNA transferred from other organisms. CRISPR does away with this. The genetic scissors can alter the plant's genome without introducing foreign DNA. The editing mechanism itself is then completely removed. The resulting corn plant, therefore, contains nothing but corn DNA. If CRISPR is used like this, the result is indistinguishable from a natural mutation. And genetic engineering, which can't be detected, is not genetic engineering, according to the US authorities' ruling on Pioneer's waxy corn.
It's not being regulated under the sort of biotech framework. It's being regulated or reviewed as a conventionally bred variety. And so, it will be launched as won't be under a GMO sort of, rubric, if you will.
A decision with far-reaching consequences. These plants will soon be growing in many fields, in the US at first. Whether European farmers will be allowed to cultivate plants modified in the same way depends on how the EU classifies CRISPR plants.
Wird die Genschere und ihre Nutzung in der
If genome editing and its use in agriculture are registered on the side of classic genetic engineering, then the use of CRISPR in agriculture is dead.
Oder wird
Or will the public accept that we really do have a veritable alternative here? That is precisely not a GMO, a genetically modified organism.
ist. Und
This is the crucial decision that divides us, that is being fought out vehemently right now, both in Germany and in Europe.
als auch in Europa gekämpft.
In the whole of Germany at the moment, there are no genetically modified plants growing in the fields. Will CRISPR change that? As yet, no one knows. And this uncertainty is a problem for European plant breeders. We're visiting Heinrich Böhm, who breeds potatoes near Lüneburg. No agro-multinational this, but a medium-sized German company. It was his grandparents who began potato breeding over 100 years ago.
Yes, these potatoes look very good.
Heinrich Böhm is not opposed to the new gene editing technique in plant breeding. He would actually like to use CRISPR. He sees it as an opportunity for combating pests and plant diseases.
One
One that gives us a lot of trouble is late blight, Phytophthora. It was actually the reason for the start of potato breeding in the 19th century.
To this day, nobody has really managed to control this disease, except with huge amounts of pesticides. In Heinrich Böhm's greenhouses, all the work is still done by hand. Different potato plants are pollinated in a bid to breed a variety that's not susceptible to late blight. Perhaps the answer lies in wild potatoes. They're tiny, but certain genes make them resistant to late blight. Using CRISPR, he could combine the best characteristics of wild and more modern varieties.
I believe that with these new techniques, we could cut the time it takes to introduce a resistance gene from wild potatoes into cultivated potatoes to about a quarter. It would mean we could introduce various resistance genes at the same time. I imagine that chemical plant protection would then be significantly reduced.
This sort of plant would therefore contribute to environmental protection. But so far, Heinrich Böhm is not allowed to use the new technique. All the seedlings here have been produced in the conventional way, which takes up to 15 years to cultivate a new variety. Heinrich Böhm has already set up a new laboratory for gene editing. He doesn't want to fall behind the competition in the US, but work hasn't yet started here. Böhm is waiting for the classification of the new CRISPR technique by the EU and Germany's federal government, as conventional plant breeding or as genetic engineering.
If it's comparable with the usual rules for genetic engineering, then it will be a technique very likely beyond our financial means. We won't manage it. So we'll be out of the running.
The problem for Heinrich Böhm is that it takes years to get a license according to genetic engineering law. It's time-consuming and too expensive for breeders like him. Then there'd be the hassle with genetic engineering opponents, though ironically one organic farmer believes that their concerns have actually created some of the problems.
In a way, the resistance to genetically engineered crops and all the regulatory steps that have been imposed have played into the hands of these large companies, because now only the large companies can afford to develop new genetically engineered crops.
Normally, organic cultivation and industrial farming are mutually hostile. At the same time, the problems are growing. The global population is increasing, biodiversity is declining dramatically. Agriculture has to come up with something. What we've seen in Davis could chart a path for the future. Raoul and his wife Pamela want to find solutions that combine the best of both worlds. For them, CRISPR is a tool to this end, nothing more and nothing less.
There's a lot of discussion of crop genetic improvement. Obviously, it's very important. All farmers rely on crops that are genetically improved in some manner, but that's just one part of farming. We also need ecologically based farming practices and stable government policies. So it's not only about genes in our food, and I think sometimes people forget that. So sustainable agriculture includes many different practices.
What role can CRISPR play, and above all be allowed to play, in solving the problems, in agriculture and in medicine? In southern Germany, researchers at the University Hospital of Freiburg are pursuing an ambitious goal, finding a cure for the immune deficiency caused by HIV. Geneticist Tony Katoman and his team are testing whether the new genome editing technique functions with precision in the laboratory. Only then can the first patients be treated.
What we are currently developing in the laboratory is a therapy for actually curing HIV. Compared with the medicinal treatment, which has to be continued lifelong, our therapeutic approach would offer a one-off treatment by which the HIV infection would be vanquished and the patient could be discharged, cured.
A dream for many HIV patients, also because a certain stigma still attaches to the disease. Dr. Susanna Uzadel is working closely with the scientists at the University Hospital. HIV is her specialist field. For over 30 years, she's been treating patients suffering from the immune deficiency.
In the 1980s, as a young doctor, I attended people who were dying, and we knew only that it was a blood-borne illness and probably a viral disease.
Since then, the therapeutic possibilities have developed rapidly. With the medication available today, many patients can lead a virtually normal life. The disease itself, AIDS, no longer breaks out, even though the patients still carry the virus within them.
This means that nowadays life is possible with HIV for a virus carrier. Now comes the cure. A lot of patients think, Dr. Uzadel will give me a tablet and that will be the last tablet I'll ever take and then I'm cured.
But it's not quite so easy, even when everything goes well. In the laboratory, the scientists are working on human cells to prevent viruses from entering them.
Every virus needs certain doors in order to enter the cells. What we're now trying to do with gene editing is to close these doors forever. The virus can't get into the cell, which makes the whole immune system resistant to the virus.
In the laboratory, this already works. The researchers switch off a receptor on the surface of the cell which the HI virus needs. They have to ensure that the genetic scissors change the genome only at the desired points. Any miscut in a cell could have dramatic consequences for the patient.
We're able to show that after gene editing, about 80% of the cells we've genetically modified are resistant to the HI virus. What's important is that we see practically no miscuts, so the precision of gene editing is extremely high.
What works in the laboratory is soon to be tried out in humans. The patients first have to undergo a course of chemotherapy. Their immune system is to be partly closed down to allow the cells modified by gene editing to spread throughout the body. Only if this functions will the patient be cured of HIV. Together with the scientists from the University Hospital of Freiburg, Dr. Susanne Usadel is preparing the clinical trial. At first, only HIV patients will be treated who have cancer and therefore need chemotherapy in any case. Despite the stressful treatment, many HIV patients have already volunteered to take part in the trial, also because they suffer from the image of their disease.
If someone has a lymphoma disease, their granny will go, "Oh my God, can I help you? Can I make you some soup and give you a hug?" But if someone says, "I'm HIV positive," have you been leading a dodgy life? Too many men? Are you gay? Or if you're a woman, how many men have you had in your life? If only we could get to where people would open up their arms when you say, "I'm HIV positive," and would ask the same questions they ask with any other disease. That would be great.
HIV patients can now live with their disease, but the stigma is hard to bear. For many, a cure is their greatest wish. But for the Rosenfeld family in London, everything depends on a breakthrough in genetic research. And soon. Time is the decisive factor for Gabriel. His muscle deficiency is getting worse, however much he fights against it.
75% of the battle is won in your head. If you think I'm going to get weaker, you get weaker. If you think I'm going to stay strong, and you stay strong. But obviously, 25% is purely what the doctors can do. But most of the fight, as I said, is in my head.
Sometimes he can hardly cope with the situation. But he has something that helps him in these moments. His mother can't stop time, but she tries her best to cherish her memories. And she tries not to think about how Gabriel's condition will develop if there's no effective therapy.
Having him in a complete state of paralysis would be much more of a fear for me than losing him. Him having a great quality of life for a shorter period of time, I can deal with. But that, living for another 20 years, unable to brush your own teeth, blow your own nose, I can't go there. So the risks I'm willing to take now for the right technology are high.
In the meantime, Ronald Cohen has gone one step further. He's telling students about success with animal experiments. The scientists have bred a genetically modified mouse, which also suffers from hereditary muscular dystrophy. Not quite the same disease as Gabriel's, but the symptoms are similar. The mouse was injected with CRISPR molecules into its bloodstream. The result, the mouse now moves almost like a healthy animal. one step further towards a therapy for human beings. But Ronald Cohn is still a long way from his goal. And time is running out for him finally to help Gabriel.
I would lie to you that there are not times where I wake up at night and wonder, whatever we're trying to do, will it be too late for Gabriel? It happens once in a while, but in general I try to keep focused, keep looking forward. And as long as we really do everything we can, then science and destiny will come together at some point.
What Emmanuel Charpentier discovered is slowly but surely changing the world. New horizons have been opened up by CRISPR, and with them, perhaps, new risks. But the crucial question is not whether we want to use the new genome editing technique, but for what? What sort of agriculture do we want? What medical therapies? And what contribution can and should genetic engineering make? If we want to shape the way things develop, we have to address the issue. And we have to do that now.
When I find myself in times of trouble, Mother Mary comes to me, speaking words of wisdom, and let it be. And in my hour of darkness, she is standing right in front of me,